Welcome back to the deep dive. Today, we are opening up the files on neuroblastoma, which is really one of the most devastating and aggressive cancers affecting young children.
It is. It's a tumor that comes from nerve cells, but they sort of, they fail to mature properly. They get stuck, essentially, in a state of just rapid, uncontrolled growth.
And rapid growth means a huge need for fuel, right? Like any fast-growing cell.
Exactly. All cancers need metabolites to grow, but neuroblastoma has this particularly strong dependency on one specific type, polyamines.
Polyamines. I know they're involved in a lot of growth processes.
Absolutely. DNA synthesis, keeping cell membranes stable, really any process that needs massive cell expansion relies on them. And these neuroblastomas are, well, uniquely hooked on making their own polyamines locally.
Okay, so if we know the dependency, there must be ways to target it. We already have something, don't we? DFMO.
Right. DFMO, or difluoromethylornithine. It's been a standard pharmacological approach for a while now.
And how does DFMO work exactly?
It's quite clever, actually. It inhibits an enzyme called ornithine decarboxylase, ODC. And ODC is the crucial bottleneck, the rate-limiting step in making those polyamines. Shut down ODC, you should, in theory, shut down the fuel supply.
In theory. But cancer's tricky.
Very. DFMO shows promise, definitely. It has clinical activity. But, aggressive tumors, especially the ones driven by really potent oncogenes, they're masters of finding workarounds. Metabolic redundancy, they call it.
So they might just make more of the enzyme or find another route.
Precisely. Or maybe even scavenge polyamines from elsewhere. So DFMO is necessary, but often not sufficient on its own for a cure.
Which brings us to our deep dive today. We're exploring a really fascinating finding from recent research, a combined strategy. Taking DFMO, but then adding something else to make it much, much more effective.
Yeah, the researchers reported profound survival gains in their models. It's a significant step.
And the really interesting part, the twist, is how it works. It seems this combined approach messes with the tumor's protein-making machinery in a very specific way, almost hijacking its genetic code.
It's a fantastic bit of scientific detective work. But before we get to the mechanism, we need to set the stage a bit.
Yeah.
Understand why these tumors are so aggressive in the first place.
Right. What's driving the malignancy?
In the most lethal forms of neuroblastoma, the defining feature is hyperactive MYC signaling. Specifically, it's the amplification of a gene called the MYCN proto-oncogene. Lots of copies of this gene.
Okay, MYCN. That's the oncogenic driver, the master switch telling the cell to be cancerous. How does that connect directly to this polyamine fuel addiction you mentioned?
It's a very direct link. MYCN, when it's hyperactive like this, essentially acts as a transcriptional activator. It turns other genes on. And what are the main genes it switches on?
Let me guess. ODC.
Exactly. Ornithine decarboxylase, the very enzyme DFMO targets. So you have this immediate tight loop. The cancer driver gene, MYCN, directly forces the cell to make more of the key enzyme needed to produce its critical fuel, polyamines.
Wow. Okay. So the driver and the engine are just instantly connected, running hot together.
Perfectly put. That's why targeting ODC with DFMO makes so much sense.
But, okay, even if you inhibit the ODC enzyme with DFMO, the enzyme still needs its raw material, right? The precursor, which is ornithine.
Right. You need the amino acid ornithine to feed into that pathway.
So where does the tumor get the ornithine from? Does it make it itself or grab it from somewhere else? Because if you could cut off the supply before the enzyme.
That's exactly the question the researchers tackled. Ornithine can basically come from two places. Tumors can synthesize it internally, usually starting from other amino acids like proline or glutamine that require certain enzymes, or they could just take it up directly from the bloodstream, from the circulation. Circulating ornithine often comes from another amino acid, arginine, being broken down elsewhere in the body.
So the tumor could be a maker or a taker. Which one is neuroblastoma?
Well, the first clue came from metabolic profiling. They looked at what metabolites were abundant inside these MYCN-driven tumors, and they found really high levels of proline.
Okay, high proline. That sounds like it's making its own ornithine internally then, right? Proline is a precursor. You'd assume it's using that pathway.
That was the immediate assumption, yeah. You'd think it's using an enzyme called ornithine aminotransferase, OAT, to convert that proline into ornithine. That's a common pathway.
But there's always a but in these stories.
There is. They didn't just assume. They used isotope tracing. This is where you label molecules, like putting a little radioactive tag on proline and glutamine, and then you track where those tags end up. You see if they actually get converted into ornithine inside the tumor.
Like following marked money through an economy, what did the tracers show?
It showed something quite surprising. despite all that proline sitting around inside the tumor, which turned out to be just uh, metabolic window dressing. Really, the tracers showed very little conversion.
So it wasn't using the proline.
Not significantly. And when they looked closer, they found that the OAT enzyme, the one needed for that internal conversion, had really low functional expression in these tumors.
Wait a minute. So the tumor has the raw materials, the proline, but it's basically too lazy or unable to make its own ornithine fuel?
That's a great way to put it. It acts like a metabolic parasite in this specific way. The conclusion was stark. These neuroblastomas are overwhelmingly dependent on just taking up ornithine and its precursor, arginine, from the circulation.
They outsource the production.
Exactly. They rely on what the rest of the body provides. And that external dependency, well, that becomes its Achilles heel.
Because now you know how to starve it. Don't just block the ODC enzyme with DFMO.
Also cut off the supply lines, restrict the main things it's pulling from the blood, which are arginine and indirectly proline, since arginine is the main source of circulating ornithine.
So that led to the combined strategy. DFMO, the drug, plus a special diet?
Correct. A novel diet specifically formulated to be free of both proline and arginine, the ProArg-free diet.
And they tested this combo in mice, the TH-MYCN model you mentioned.
Yes, that model is designed to mimic the aggressive human MYCN-amplified disease very closely. They gave mice the control diet, the ProArg-free diet alone, DFMO alone, or the combination.
What were the results? Did the combo work?
It worked remarkably well. The diet alone, just a minor effect, barely slowed things down. DFMO alone. It definitely extended survival compared to controls, but many tumors eventually relapsed.
That the combination.
The combination therapy, the diet plus DFMO, induced a really marked, significant survival benefit. We're talking about durable responses. Around 20% of the mice treated with the combo remained completely tumor-free, long-term, even after the treatment stopped.
Wow, 20% cure rate in a model this aggressive that's substantial.
It really is. It showed the synergy was powerful.
And did they check the metabolism? Did it actually deplete the polyamines more effectively?
Oh, yes. They measured the levels of key polyamines, like spermidine. The combination treatment caused a dramatic drop, more than a 10-fold reduction compared to controls. It was significantly better at depleting polyamines than even DFMO, combined with a drug designed to block polyamine uptake.
So starving the precursors and blocking the enzyme really did shut down the fuel production line.
Absolutely. It proved the concept metabolically. They hit that pathway hard from both ends.
Okay, so we've got a therapy that works much better, induces cures in some cases. But here's where it gets really deep, I think. You mentioned it wasn't just killing cells. Some tumors seem to be reprogrammed, turned into something less dangerous.
That's right. The long-term survivors often had tumors that had differentiated, becoming more like mature, benign nerve cells instead of rapidly dividing cancer cells.
Check.
Starving them didn't just kill them, it changed their fundamental nature. The question was how? How does this metabolic stress trigger such a profound genetic reprogramming?
That's the million-dollar question. It feels like we're moving beyond just cutting fuel lines now.
Exactly. This is where the investigation shifted from metabolism to the actual process of building proteins translation. To see what was happening at the molecular level, the researchers employed a technique called ribosome profiling, or ribosec.
Ribosec. That sounds like looking at ribosomes, the protein factories.
Precisely. Think of it like taking an incredibly detailed snapshot of every single ribosome on every single mRNA blueprint in the cell at the exact moment you freeze everything. It tells you where the ribosomes are cruising along smoothly, and importantly, where they are pausing or getting stuck.
Okay, and we know polyamines are generally needed for translation, right? There's this factor EIF5A that needs polyamines to work properly, a process called hypogenation.
Yes, that's a known link. Polyamines, particularly spermidine, are essential for modifying EIF5A through hypogenation. And properly modified EIF5A is crucial for smooth translation, especially around stop codons and certain tricky sequences. So the first hypothesis might be polyamines drop, EIF5A function fails, translation gets generally messed up.
Did they see evidence of that general breakdown, like problems at stop codons?
They did see some signs consistent with EIF5A dysfunction. They observed ribosomes tending to accumulate near stop codons, which fits the picture. And they directly measured EIF5A hypogenation and found it was reduced in about half the tumors under the combination treatment. So that part seemed to be happening.
But I sense another but.
But the ribosec data revealed something far more specific, far more targeted than just a general translation mess. The ribosome stalling wasn't random or global. It was highly codon specific.
Codon specific. Okay, hang on. A codon is the three-letter RNA sequence, like CCA or CCG, that tells the ribosome which amino acid to add next. An amino acid like proline can be coded by several different codons. You're saying the ribosomes got stuck only on certain codons, even if they coded for the same amino acid?
Exactly that. It was quite remarkable. For example, proline can be coded by 4 codons: CCC, CCU, CCA, and CCG. What they saw was significant ribosome stalling predominantly at just one of those, the CCA codon.
Just CCA, not the others.
Minimal stalling at the others. CCC, CCU, CCG were largely read smoothly. This couldn't just be because the cell was running out of proline, because then all proline codons should stall. And it wasn't just general EI-F5A failure, or that would likely affect translation more broadly, perhaps at specific motifs like polyproline sequences, not just one specific codon.
Wow. That's an incredible level of specificity. It's like the ribosome hits a very particular speed bump, but only when it reads CCA. So what was the pattern? Did they figure out a rule for which codons were causing the stalling when polyamines were low?
Yes, they looked globally across all 61 possible sense codons. They found a striking pattern related to the third nucleotide in the codon, what's often called the wobble position.
The wobble position, that's the one that often varies for codons specifying the same amino acid, right? Due to flexibility in tRNA pairing.
That's the one. And the rule they uncovered was this. Under the low polyamine conditions caused by the combined therapy, codons ending in adenosine, A-ending codons, showed a dramatic increase in ribosome occupancy, meaning ribosomes were stalling or pausing much more frequently at A-ending codons.
A-ending codons specifically, so things like CCA for proline, GCA for alanine, ECA for serine, those became problematic.
Exactly. Those showed increased pausing. And conversely, codons ending in guanosine, G-ending codons like CCG or GCG, actually showed reduced pausing. Ribosomes seemed to move through them even faster, perhaps.
That's wild. A previously unknown metabolic link where depleting polyamines specifically gums up the works for reading A-ending codons. Why A? Any ideas on the mechanism?
It's still an area of active research, but the leading hypothesis involves the transfer RNAs, the tRNAs. These are the molecules that actually bring the amino acid to the ribosome and match the codon.
Okay.
Many tRNAs undergo complex chemical modifications, especially at position 34, the part that directly interacts with that third wobble codon position. The idea is that polyamines might be essential for either making or maintaining specific modifications on the tRNAs that read A-ending codons.
So without polyamines, those specific tRNAs can't bind efficiently or correctly to the A-ending codon on the mRNA blueprint.
That seems to be the most plausible explanation. The tRNA responsible for reading TCA, for instance, might need a polyamine-dependent modification to work properly. When polyamines disappear, that tRNA becomes inefficient and the ribosome waits. It stalls.
So it's a targeted disruption of specific tRNA functions, leading to these codon-specific delays, a metabolic state controlling translational speed based on codon choice.
Precisely. A metabolically induced, codon-specific speed bump.
Okay, now I connect it back to the cancer outcome. If the cell is suddenly bad at translating A-ending codons, what happens to the proteins whose blueprints are full of those codons?
This is where they integrated everything. The RNA levels, what blueprints are available, the ribosec, where the factories are stalling, and crucially, proteomics, which proteins are actually getting built.
Proteomics measures the final protein output.
Right, and they saw clear changes at the protein level driven by the combination therapy, even when the RNA levels for those genes didn't change much. This confirmed it was a translational effect, a disruption in protein building, not gene expression.
And did the protein changes match the A-ending codon rule?
Perfectly. There was a stark contrast. The protein sets that were most strongly downregulated, the ones whose production was most effectively shut down by the therapy, were genes involved in the cell cycle.
The very program driving the cancer's aggressive proliferation.
Exactly. Things like CMPR, KUF2C, CEP57, key components of the cell division machinery. And when they analyzed the codon usage of these genes,
Let me guess. Loaded with A-ending codons.
highly enriched, dramatically so. For example, they mentioned the CEPR gene, critical for cell division. In the human transcriptome, the average gene uses A-ending codons about 18.8% of the time. CEPR. It uses them 38.6% of the time.
Wow. So that gene was basically encoded to be incredibly vulnerable to this polyamine depletion strategy. It's like it was written in a language the cell suddenly couldn't speak fluently anymore.
That's a perfect analogy. The proliferation program was effectively silenced at the protein level because of its inherent codon bias.
And the flip side, what about the proteins that increased, the ones driving that desirable differentiation effect?
Those were genes associated with neuronal differentiation, the pathways that make neuroblasts mature into proper nerve cells. These protein sets were the most upregulated by the therapy.
And their codon usage.
Critically, these beneficial differentiation genes were found to be depleted in A-ending codons. They used fewer of the problematic codons.
So they were spared. The stalling mechanism didn't affect them as much, allowing their proteins to be made successfully and take over the cell's program.
Precisely. The therapy inadvertently created a selective pressure. Suppress the A-rich proliferation program, allow the A-poor differentiation program to emerge.
Okay, let me try to summarize the whole mechanism then. The combination of DFMO plus the proarg-free diet creates extreme polyamine depletion. This depletion doesn't just cause general chaos, but specifically messes with the reading of adenosine-ending codons on the mRNA, probably via tRNA modifications. Because the cancer's key go-go-go cell cycle genes happen to be packed with these A-ending codons, they get translationally suppressed. Meanwhile, the slowdown in mature neuronal differentiation genes, which use fewer A-ending codons, are allowed to be translated, effectively forcing the cancer cell to reprogram itself into a more benign state.
You've nailed it. That's the core discovery. And the visual proof came from histology, looking at the tumor tissues under a microscope.
What did they see?
The tumors treated with the combination therapy showed clear signs of differentiation. Cells had more cytoplasm relative to their nucleus. They developed abundant neuropilatote, the network of nerve fibers characteristic of mature neural tissue. It was morphological proof of successful reprogramming from aggressive neuroblasts to something much more stable and differentiated.
And did this hold up beyond the initial mouse model?
Yes, importantly, they replicated key aspects of this, showing durable responses using the combined therapy in human neuroblastoma cells grown as xenografts in mice. This suggests the mechanism is conserved and clinically relevant.
This is really fascinating. It adds a whole new layer to how we think about metabolism in cancer, connecting diet right down to the specific letters in the genetic code being translated.
It really does. It reframes codon usage, which scientists often thought of as somewhat redundant, just different ways to code the same amino acid as a potential regulatory system linked to metabolic state.
Let's just quickly recap the key takeaways for everyone listening. What are the absolute must-remember points from this deep dive?
Okay, 4 main things stand out. 1, neuroblastoma, especially the MYCN-driven kind, heavily relies on external arginine and ornithine from the blood to make its polyamines. This makes it vulnerable to restricting those amino acids in the diet.
Right, the taker, not the maker. Second point.
Second, combining that specific dietary restriction, the proarg-free diet, with the ODC inhibitor drug, DFMO, achieves a much more profound depletion of polyamines than either approach alone. You starve the supply and block the factory.
Got it. Third,
this extreme polyamine depletion triggers a very specific, unexpected consequence at the ribosome level, stalling or pausing, specifically when the ribosome encounters an adenosine-ending codon on the mRNA blueprint.
the A-ending codon rule.
Yeah.
And finally, the fourth point.
Fourth, this selective stalling mechanism hits the cancer where it hurts. The aggressive cell cycle and proliferation genes are disproportionately enriched in these A-ending codons, so their translation gets suppressed. At the same time, the neuronal differentiation genes are depleted in A-ending codons, allowing them to be translated, which pushes the tumor towards a benign differentiated state.
So the therapy essentially exploits a hidden metabolic vulnerability encoded directly into the codon usage of the cancer's core programs.
Exactly. It suggests that maybe these codon usage patterns didn't arise randomly. Perhaps core programs essential for rapid growth, like the cell cycle, evolved to use these metabolically sensitive A-ending codons almost like a built-in safety switch.
A switch that allows metabolic stress, like resource scarcity, to quickly and coherently shut down growth and maybe trigger an alternative program, like differentiation.
That's the intriguing implication, that metabolic state and codon usage could be intertwined regulatory systems. And in this case, researchers cleverly hijacked that natural system to force a therapeutic outcome in a very dangerous cancer.
Which leads us beautifully to our final provocative thought for you, our listeners, to ponder. If a cancer's fundamental growth program can be encoded in its preference for certain metabolically vulnerable codons, like these A-ending ones,
Then what other non-essential dietary components might be interacting with codon usage biases in other hard-to-treat cancers? Could specific dietary choices be subtly fueling aggressive programs, not just by providing raw energy, but by enabling the translation of proteins whose blueprints rely on specific codons sensitive to those nutrients? It suggests diet might influence not just the cancer's fuel, but the very language it uses to build itself.
a fascinating question to consider. That's all the time we have for this deep dive. Thank you for joining us.